guys have spent a whole bunch of time at this conference talking about the relationship between humans and nature, right? It's the concept of ethnobiology, and this has both environmental and evolutionary implications, not just in the physical, but as well as in the psychological conception that we have. Our perceptions of our ecosystem are also affected by this relationship between them. And interestingly, one of the things that a lot of people, when they're studying this relationship between man and nature, plant domestication is a common area that's studied in this realm. But when you talk to people, I've tried this a lot in classes, I said, ask my students, how would you define nature or natural? And the vast majority of the time, the answer that I get back is something along the range of a natural area is an area that is not touched by people. So there's this automatic implication or bias that humans are not part of the natural system. But this is actually something that was really common in ecological thinking for a very long time. But recently there's been a few perturbations to this idea. So Nietzschean theory is an ecological theory looking at the idea of how humans or how organisms interact with their environment. So the idea is that organisms don't only adapt to their environment, but they in fact alter their environment to adapt it to their needs and their resource needs. And so this has actually been adopted by Smith to look at initial plant domestication and the beginnings of agriculture, and saying that in fact this is a form of niche construction where humans have actually adapted their environment and the organisms in them to help provide for their resource needs. And so obviously this is going to vary in intentionally and as well as in direction, but the idea here is that when you think about how this creation of a niche, well, it's not an instant thing, it extends on through time. There is a temporal implication of this. And so Ballet, when he was talking about the research program of historical ecology, he said that past events have been inscribed sometimes subtly on the land. And it's interesting to start thinking about what is meant by subtle. I mean, how subtle is it? And when you're thinking about time, how much time does it take to wash away these inscribed events that have happened? And it would often depend on what kind of selection or management we're talking about. Now, when Darwin first introduced the idea of artificial selection, he actually broke it into two different types of selection. So first there's conscious selection, which is one we're most familiar with. So this is intentional selection of breeding individuals, and then hence the assumed destruction of all the other ones. This is mostly done with our common annual weedy species that are common domesticates like corn. And so the idea is they have these short lifespans and they're quickly moving into reproduction. And so they were breeding individuals were selected, everybody else was eaten, and they planted for the next year. So this has very quick and pretty obvious ramifications for domestication syndrome creation for a lot of these organisms. But there also is, I keep hitting this thing instead of using this thing in my hand. Are you noticing this? But there's also unconscious selection. Now in unconscious selection, Darwin said that this was when you had preservation of useful individuals. So not necessarily selection of breeding individuals, but you're preserving some and destroying the others. So for example, when you were clearing a field, you would leave the best fruit trees there and then cut everything else down. And so obviously this would have some evolutionary ramifications. But so Hari actually expanded this idea to say that there's actually environmental impact here, because when you do move something from the wild into an anthropogenic setting, then this is also going to affect its potential evolutionary future. And so the result of either of those is going to be evolutionary change. Now, when we're talking about perennial species, this is a little bit more difficult to see these inscribed events in them. So a lot of our fruit trees, our fruit crops, are actually dioecious species. Now, by dioecious, I mean they have separate male and female individuals. This is actually pretty rare in the plant world. It's about 6% of angiosperms. And the idea was that they theorized that this evolved to maximize outcropping amongst individuals. And because of this situation of having separate male and female individuals, as a farmer, this is not terribly convenient for you. So if you have a whole bunch of males that you just spent 5 years growing to reproductive maturity and then wham, no fruit. So for them, it made a lot more sense to primarily use vegetative propagation for these. But this has actually resulted in a high maintenance of genetic diversity in a lot of these perennial fruit crops. And so a recent review on perennial fruit crops has shown that on average is about 95% of genetic diversity in fruit crops as compared to their wild relatives. So it's a really high level of diversity maintained. But this also leaves us with something of an unclear domestication syndrome, such that you have some of them where it's actually very difficult for people to figure out whether there really is a separate domesticated species or not. But there actually has been a lot of recent research in a lot of traditional communities showing that in these long-lived perennial crop species, there are actually other management options that are used that do actually lead to evolutionary change. So they do things other than this cloning. And so Casas et al. In the lab down there does a ton of work in Mexico on in situ traditional management of perennial crop species. And he said, aside from the let standing idea that we mentioned, well, you clear the field and you leave a few individuals that are very useful, aside from that, there are also management techniques like encourage growing. So perhaps you remove competitors from its area or dump all of your food garbage around it to give it a little bit of a boost, an organic fertilizer there. Also protection, which seems relatively obvious what that might mean, and then systematic gathering. So you can help then determine both spatially as well as temporally perhaps on where these organisms are and then which groups within different populations are actually going to have more reproductive opportunities. Okay. So, and what's been shown is that all of these different land management techniques do in fact lead toward incipient domestication and evolutionary change. Now obviously all of these are going to be varying in intentionality and intensity. But Zohari, when he was looking at this, actually said there actually, when you're talking about these perennial crop species, there tends to be two outcomes. Either the plant just pretty much decides, being anthropomorphic, the plant decides that it's forget it. This whole fertilization thing, it's for the birds. I'm skipping it, I'm going straight to making my own fruit without help from anybody else, and they go to parthenocarpy. Or the other option is that they move away from dioece and move into monoece, meaning an individual that has both male and female flowers on it, or you have a situation where some plants will develop hermaphroditic flowers. All right, now this obviously is something that, in order to get to this level, you need consistent management over time. But this comes back to this idea of inscribed events. So after something has been managed consistently for a long period of time, what happens if it's abandoned? If the culture that managed it for one reason or another stops, or they disappear? So it's not like the plants simply disappear with them. So what happens at that point? And so this was kind of what I thought was rather interesting. On these historically managed species, what would be the result of this long-term abandonment? So the species I'm talking about today is Diospyros virginiana, the American persimmon. Now Diospyros means food of the gods in Greek. And this actually, if you think about it, Diospyros kaki, the Asian persimmon, is arguably the most popular fruit in the world. Bet you didn't know that. But weirdly enough, the American persimmon, very few people even know about the American persimmon. Down here it's a little bit more common, but even so, it's just a wild weedy thing that wild harvesters might collect occasionally. But it has a really rather huge range throughout most of the southeastern US. And it is a dioecious species, and it's listed as such in literature, but there is anecdotal and observational information in the literature from the early 20th century saying that incidences of monoece, gynomonoece, which is female plants with hermaphroditic flowers as well, and the opposite being andromonoece, which is a male plant which will have a branch or two of perfect flowers, have all been observed, as well as parthenocarpic individuals. Now, of course, this is a little bit fuzzy because these are very anecdotal. It was hard to say whether these were cultivated individuals or not, the ones that were being described. But all of these have been observed, and it has this really bizarre reproductive system. And so this species does have a very actually long history of use by people. And so it was mentioned very extensively in early writings when Europeans first started moving over. They mentioned the use of American persimmon by indigenous cultures in the area, as well as early settlers used it a lot themselves. And it's regularly found in archaeobotanical remains from, for a very long time. So just a few examples of this from the later Archaic, in a series of sites in the southeastern US, they found that in many of these, it's actually the most ubiquitous fruit crop found in the archaeological series. And then in Mississippian sites, Scarry found that it's actually a component in more than 80% of midden pits. And so it's very common, and it's very heavily used historically. And then even there are also historical records of orchards that were remnants of orchards that were found by the Europeans when they showed up. The Native Americans having been having a population decline upon the arrival of our European brethren, their populations dropped. A lot of these areas were abandoned, but Europeans report these former orchards that had a lot of fruit species in them, including the American persimmon. And so my question is, can we still use, can we still detect these imprints of human management after 500 years of essential abandonment and where the species has been left to be feral in the forest? And so maybe it's possible that this can actually be true. There is a case of Rosamund Castrum. Any of you that have worked much or watched any of the talks today about the Maya in Latin America might have heard of this species. So Poulston, in his dissertation, actually expanded this, brought in this idea that the high density of this tree species, which is commonly known as ramon, its fruit is a nut-like that's very high in nutrients. And he said that the high density around Tikal, Guatemala, which is an ancient Maya site, was actually due to Maya management. And this has expanded into a pretty enormous theory. And it became very controversial in the archaeological world for a while. Labert and Arneson wrote a paper refuting that, saying, well, in fact, it's due to bat behavior, how bats make use of these ruins for roosting, and so they bring in all these ramon seeds, and all the seeds get dumped out when they poop everywhere, and voila, we got ramon all over in these sites. But Peters made just an observational paper on some notes, and he said, well, in fact, I'm looking at the population of Rosamund castrum around Tikal. He noticed a difference in their phenology from other Tikal populations in similar forest areas, that they actually flower more times per year, and he saw instances of changes in the breeding system from Dioece to Monoece. So it's a hint, maybe. So, of course, we immediately run into big questions. When we're having something over a long period of time, there's a lot of potential confounding variables, and the first one that came to mind was edaphic variability, because of the fact that persimmon has such a huge range. It covers a lot of different ecotypes. And one common response to species that move across many different ecotypes is what's known as spatial segregation of the sexes. So due to variance in nutrient needs between males and females, then you might actually expect to find them living on different soil types. And so an example of this in Diospyros is actually, there was a study in Mauritius where looking at 11 different Diospyros species, and they found that there was an instance of leaky Dioece, which was found to be a species that grew in very poor soils. And so they suggested that the leaky Dioece was actually due to this environmental heterogeneity. And like I said, virginiana very tolerant of different soil types, so maybe that's what's going on to some extent. And then the second problem with it is that in the literature there was mentioned that there are two karyotypes of Diospyros virginiana. There's actually a tetraploid and a hexaploid karyotype within the species. And this was suggested that there's actually a geographic component to this, that the hexaploid is on the periphery of the range, on the north and the west edges of the range. And suggested maybe this has something to do with cold tolerance or drought tolerance. Not really sure. Now this was done in 41, and so this is based on chromosome squashes, which are a little bit fuzzy sometimes. And the big issue with this is a very limited sample size, and of what they did have, there was inconsistencies. in it, and most especially being several 2N90s that were smack in the middle of the 2N60 range. So a little unclear what's going on there. I did it again. All right, so first step was trying to get at some of these issues, these confounding issues. So what I did was the big issue of trying to attack this is how to get, I needed a large sample size, obviously, because of the extraordinary range of the species. Well, and so I went to herbarium specimens, because these things obviously have been collected in North America for quite some time, and it covers the entire range. And so I got these specimens, I had 31 males and 47 females, because I needed to know what sex they were in order for me to plot these out and see if there was any relationship to gender in terms of their relationship to soils. So these guys ranged in age quite extensively. So some of them were older than 100 years old, whereas some of them were relatively recent. And so for all of these, there was either GPS coordinates, which were collected at the time that the specimen was collected, or if there was, the older specimens just had a description, which we then geolocated using GIS. And so you might not know this, but the federal government actually has a database where soils from every state are submitted. There's required to have a GIS database from every state on their soils. And then these are often quite precise. You can get these 10 by 10 quadrats throughout the whole state. So you can actually get some very specific information. What you don't get, however, is much consistency. So what things we could find that were consistent between all the states were pH, cations, annual precipitation, and slope. So those were the edaphic characteristics that we used. And so based on our GPS coordinates, we actually found those, the data for all those variables in those exact locations. All right, so we also wanted to look at karyotype analysis, right? So first idea was to try flow cytometry. This is a very powerful method of investigating ploidy in plants, and it has been reported in literature to be useful when using dried samples, herbarium samples. There have been quite a few papers talking about, oh yeah, you can totally do this with herbarium samples. Not. Turns out that this really only works when you have a relatively young herbarium sample. So more than a year, everything kind of falls apart because they just get too dry and you can't find intact nuclei. So after fighting with that for a while and completely failing, I had another brilliant idea, which there was also in the literature info about stomata length, where actually stomata length, the garden cell length, should actually correlate with ploidy in plants. And so this has actually been used in many species to distinguish ploidy. And so went and painted my leaves with clear nail polish, dried packing tape, pull it off, stick it on the slide, and did this 350 times so that I had enough samples for everybody, and then went in and on a scope and tried to measure stomata. And after enormous numbers of hours of this, another. So, you know, hey, I gotta give me points for trying, right? But what we do have, so here is our distribution map. So all of those are the specimens mapped on there. You can see the pluses are males and the circles are females. And so this pale gray area is the USDA boundary for the species. At the top are basically the extent of the last glacial maximum. So the dark is where the actual solid ice was, and in front of the hatch marks, that's where the polar desert that comes off of the edge of the glacier was. And you can see it actually, the species very closely hugs up to the Wisconsin glacial. And so when we looked at the edaphic variables, we looked at this a couple different ways. First, I did just a simple NMDS, non-parametric ordination, just to look, it's basically a way of exploring your data and observing, hopefully relationships. And then finally, I did an analysis of similarities, which is a non-parametric version of ANOVA, looking at comparing variation both within and between your a priori defined groups, and in this case, it was gender. And so what I found, no, there is no difference in, as far from our data anyway, on the soil preferences of males and females in this species. So the NMDS, you can see pretty much total overlap between males and females. Again, males are crosses, females are the circles. And the Anderson, we get a p-value of 0.92, so not any difference. So no spatial segregation of the sexes according to that. And like I said, I did do the stomatal peels, I wasn't kidding. And so here you can see, again, in NMDS, looking at males and females heavily overlapping. And then I thought, well, maybe it has to do with area, right? You know, like where location. And so I just thought, you know, I'll just look at this by where these things are at, right? And so just to give you an idea about what might have been going on here, if you look at just the dark X's, which is Ohio. Ohio is the edge of this range of this species. And what you can see is those dark X's are scattered all over the place. The variability in this is so high. And that could simply be just because my eyeballs were getting weird after measuring all these, or I mean, it's kind of hard to say. But you know, there's a lot of variability, so couldn't really tell much from the stomatal length. So obviously, I am going to want to continue on with doing the flow cytometry in the future when I get some fresh specimens. And so, like I said, flow cytometry in the future. But also, our plan is to do some both field and common garden trials, looking at these guys in the core of their range as well as on the edges of it. And then possibly anything else. Anyone has brilliant ideas, please let me know. But the interesting, I mean, I think this species is really cool and potentially valuable case study that, that we're going to keep pursuing. And so it's really getting us into this idea of plant evolution and ecology, looking at this within a historical framework. And we can look at this at different scales, population, ecosystem. We can also have these different perspectives because this is an inherently interdisciplinary study. And so it gives us a lot of opportunities here. And you know, aside from the influence, the anthropogenic influences, this really gives us kind of interesting approach to looking at evolutionary mobility, dioecious systems, and as well as maybe potential for adaptive flexibility in the reproduction of these dioecious species. And so theoretically, these should be good for not only just really cool botanicalness, but also just conservation and, again, you know, looking for new native crop species. So this is a species that has a lot of potential in a lot of different ways, and it's just cool. All right, so thank you very much. Have a question?
Yeah. Were the stomatal measurements a failure because they didn't show ploidy differences, or because there wasn't a pattern that came out of it?
Well, because there wasn't a pattern. I mean, because I was hoping to use them. I had control. I don't know if you noticed that, just that of a known ploidy, but those two were scattered all